Amazing. Well, good morning, everybody. I'm actually surprised to see an audience this broad. I was actually looking forward to speaking to the four people who made it 10 minutes before the start. Well, my name is Kristina. Thank you very much to the organizers for having me here today. It's a fantastic pleasure to join New Bias and May, and now the CBIAS, which is growing in numbers. And it's absolutely amazing to see the rise of the community and how many people are actually interested in bioimage analysis. So today I'm going to spend 25 minutes talking to you about how we learned dynamic image representations for singles for cell cycle annotations. I'm going to try to make a point about how we approach this task in an unsupervised or self-supervised way. So for the supervision enthusiasts, hopefully you'll find it useful. I try to think of myself as a temporal person, so I'm very interested in changes and processes over time. So hopefully if you're interested in tracking, or if you're interested in dynamic processes that evolve and develop over time, hopefully you find the talk useful. But before the talk, I was actually tasked with a very amazing task from the organizers, and they asked me to actually introduce the institute, which I'm joining you from. So I work at the Alan Turing Institute, which is, I actually don't know which way, but somewhere across the street from the Crick. We're actually based in the British Library. So let me just spend a slide or two just telling you a little bit more about what the Alan Turing Institute is and what it's all about. So the Turing Institute has been founded in 2015. And since then, it has been profiled as the UK's National Institute for AI and Data Science. And hopefully that subtitle kind of tells you that the main interest of the Turing Institute is to tackle challenges in terms of data science and artificial intelligence. I would say the motto of the Institute is to have impact at scale and to help with progress against societal challenges. And the societal challenges aren't necessarily limited just to bioimage analysis in terms of microscopy, which is the field that we've been mostly discussing here at this conference. But the Turing actually has 3 grand challenges, and the grand challenges are separated or defined as follows. So the first, or the largest challenge that the Turing is interested in, is the defense and national security. So the Turing actually acts as a UK government advisory body, and it's trying to address this goal to protect the UK, its people, and the places they inhabit. The second grand challenge that the Turing is very interested in is the environment and sustainability, with the motto to addressing the climate and biodiversity crisis and the increasing need for sustainability in our developments and our daily lives. And the third one is the transformation of healthcare. But however, the transformation of healthcare doesn't just apply to the UK. It's actually aiming to transform the healthcare and enable better outcomes for all. And it's probably not surprising that at the bioimage analysis and microscopy analysis conference, I am the person who is mainly involved in this particular challenge. So I actually work on a sub project or a project that is called Building the Google Maps for Molecular and Cellular Biology. You probably heard this name or a variant of this name yesterday in Tong's presentation, and that kind of suggests that the Turing Institute is very interested and welcoming collaborations from other institutes around the UK, but also around the world. We're currently working on many different collaborative projects with the Sanger Institute, also with the Crick and many other places. So if you're interested in getting in touch with the Turing, please do let me know, and I can definitely point you to the right people to get that going. So what is when I was actually getting these slides ready, I was actually reading through the description of what is on the website regarding the Google Maps project, and one of the goals of this project was to provide an unprecedented view of the living matter organization. And that really resonated with me, and it kind of clicked with me because during my entire PhD, I was really interested in how the living matter is organized and how we can understand how cell populations develop and what they're doing in natural populations. So I was interested in cell culture, and so for me, it was a very natural move to now transition to the Alan Turing Institute, where I'm doing my postdoc. So let me just transition to what I'm actually interested in and how, my topic of the PhD kind of naturally led me to join this transformation of healthcare grant challenge at the Turing. So during my PhD, when I started, I was actually very interested in measuring single cell trajectories. And what I wanted to do was actually measure biology. I was a first-year PhD student. This is probably a very naive task, but I really wanted to measure biology. And what I meant by it was to be able to quantitatively annotate and quantitatively follow single-cell trajectories of cells growing in culture. What I mean by that is that we have a fantastic repository of time-lapse microscopy data, as you can see on these slides everywhere around. And as you can see, we capture these cells growing in cell culture, growing in a population with a relatively short time, in a relatively short time interval. So we take a snapshot of these cells every 4 minutes, and what happens here is what happens here is that we can continually we can follow these cells through divisions, and we can follow these cells over multiple generations in cell culture. What I really wanted to do during my PhD was to able to devise a set of automated tools that can help us translate this set of images into automatically extracted tracks. So to be able to follow a single cell from its birth to its subsequent mitosis, and to actually assign the children of that cell to the parents, so that we can follow what is happening over the growing generation in a tree. So to do that, we, as I mentioned, we started doing live cell imaging experiments. And when I say we, I should definitely give a shout out to the postdocs who actually took all of this data in the lab. And the data were comprised of the MDCK cells. The MDCK cells represent, as normal as it gets, a healthy human epithelium. So these are kidney cells, which have been transfected with 2 markers. One of them is an H2B marker, so that we can see where the nucleus of every cell is. And we also transfected these cells with PCNA marker, which tells us quite a lot about the cell cycle progression. So as you can see, this is what the fluorescence images would look like. As you can see here, the time frame, as I mentioned, was relatively short. So we would take a snapshot every 4 or so minutes, and we would actually continue an experiment for a couple of days. So for 3 to 5 days, which actually helps us acquire a lot of fluorescence images, and therefore a lot of data. We ideally we would want to get from an imaging movie like this, we would want to get to an automatically reconstructed lineage, and that's exactly what I'm showing you here. So a lineage, or a lineage tree representation, actually captures every single cell which we captured in this movie, and we would follow that cell over generations. We would follow its progeny and how quickly or how slowly it divides. So I don't want to spend too much time trying to explain how exactly this pipeline works, but I do want to show you what the output or the result of such a pipeline would be. So here I'm going to show you a 12 seconds of pure beauty. This is one of my favorite videos that I've generated during my PhD. And for your convenience, what I'm showing you here is that I highlighted in 3 different colors. I highlighted 3 cells, 3 clones, which will have a lot of heterogeneity in them. So just pick one and follow in this video how these cells are dividing. What you see here is a segmentation mask of all cells, and you'll actually see that even though we're starting from a relatively sparse population at the beginning, we're actually going to get to a relatively dense and confluent monolayer towards the end of the movie. So enjoy. So even by now, you can hopefully see that the cells actually behave very differently. There are some cells that are dividing very slowly. They barely go through 1 or 2 rounds of divisions. However, there is a cell, especially the green one, which seems to be very aggressive and actually divides very rapidly. I'm going to play this again just because I really like this movie, and it's just 12 seconds. I always have 12 seconds to spare. So let's just see if you can follow a different cell for now. Cool. Silence. Okay, I really hope that you enjoyed this video, because what the video kind of shows you what I'm trying to the point that I'm trying to get to, which is, even though we have a very homogeneous population of cells growing under tissue culture, we can see that there is a massive heterogeneity in the cell cycle and in the cell cycle duration, but also in the mitotic activity of those cells. That doesn't really make much sense, because you would assume that these cells would not be as heterogeneous as these, because there is no drug perturbation or no sort of drug uptake or any sort of genetic knockdown or knockout introduced to those cells. They're supposed to be as homogeneous as possible. But as you can see, this is not necessarily true. The cells are behaving very differently, and so this was a puzzling question for us, and especially to me when I was doing my PhD, I was really puzzled by why this heterogeneity arises. Now, when we, took these movies and we actually were able to devise automated tools to extract these trees, we try to actually have a look at these trees and spot whether we can assign some sort of reason for this heterogeneity in the data. And the tracking was absolutely great because it helped us to automatically extract two things. First of all, it helped us to extract how long each trajectory is, so that we can plot a distribution of all of our cells and all of our data in these movie repository. And as you can see, we could kind of recover a histogram looking just like this, which kind of shows you the distribution of the cell cycle durations. The puzzling thing here is that there's most of the cells divide for about 15 to 20 hours or in that particular range. However, there are some cells in that population that divide for as little as 7 or 8, whereas there's also cells that take almost 40 or even more than that, 40 to 42 hours to divide. So the question that we started asking here is, okay, can we actually understand how this heterogeneity occurs? And one very natural thing that we tried to do was to have a look at these trees and understand whether there is some sort of heritability or heritable propagation of the cell cycle duration in these trees. And we got to a certain extent with the, with the analysis. We actually could devise that there are some relationships between the mother and the daughter cells. However, we weren't actually getting very close to what the cell is doing. And so what I became quite interested in is that I told you at the beginning that I wanted to actually measure and quantify biology temporally along these single cell trajectories. So a natural question for me to ask was, can I actually temporally label these trajectories? And can I start asking myself whether there is a particular duration of the cell cycle phases like the M, G1, S, and G2, and whether I can somehow start understanding what is going on in terms of the cell cycle. There has been some evidence in the literature that it's actually the length of the G1 phase that dictates the length of the entire track. And so I had a very simple question to ask myself: is there a correlation between the length of the G1 phase and the length of the entire track? Now, this question might seem a little trivial, or it definitely sounds like it should be easy to answer. However, it's much more complicated than it actually sounds. Now, the reason for that is that when we look at these single cell trajectories, they're not as easy to annotate and as easy to label as it may sound. This is because biology is full of continuous transitions. And even though I tried really hard to take a couple of dozens of these trajectories, and I tried to label where exactly it is that the G1 phase and the other phases start and finish, I wasn't able to do that. And that is because biology is full of continuous transitions. There is not a certain cutoff where people can definitely say that, okay, well, this is the start, this is the first frame that the cell is in G1, and this is the absolute end. There are some transitions that are sharper than the others, but usually this is a very complicated problem to solve. The other problem that we had was that when I wanted to understand and compare these trajectories one to each other, this was not a trivial task either, because all of my trajectories were of a different length. And what I mean by that is, hopefully I try to illustrate it here in this figure. What I mean by that is that because they live for a different number of frames, it's really difficult to compare them. And it's really difficult to compare them on a frame-wise basis, but it's also difficult to compare them in terms of the length, the relative lengths of their phases because of this heterogeneity. So all of a sudden, even though I had a simple question to answer, I had 3 challenges that I had to fight against. The first one was that the cell tracks were of unequal lengths, and that really complicates the comparisons. The second one was that the cycle phases are actually variable in durations themselves. And the third one was that the trajectories do not have these sort of sharp transitions, which are very easy to annotate. Let me just show you what the real data looks like, because I feel like there was enough of these schematics. To be able to answer the questions and to be able to actually, start bringing the real data into the picture, we are now dropping the histone marker, which is really helpful for localization of the cells, but it doesn't tell us very much about what the cell is doing in terms of its cell cycle. What we're going to do here is that we're going to have a look at the PCNA marker, which is the marker that we used especially for the purpose of understanding what the cell is doing in terms of its life cycle. And as you can see, the PCNA is very helpful for us because the PCNA is actually able to tell us when these transitions between G1, S, and G2 phase happens. I don't know if you can actually see from the slide. It kind of points out to the fact that we were solving and dealing with very noisy data. But hopefully, you can you can believe me that there is a very smooth transition from mitosis to G1 phase, and then in G1 phase, the pCNA marker starts to accumulate these clusters or granules or speckles, no matter how you'd like to call them. After the G1 phase is finished, the speckles all of a sudden they disappear from the nucleus because the pCNA gets recruited to help with the DNA replication machinery, and all of a sudden you have a very distributed signal. After the distributed signal, once the S phase is finished, as I was saying, the transition from the S phase to the G2 phase, again, is very smooth and gradual, and it's very difficult to pinpoint the transition to the particular frame of where exactly it happens. After the G2 phase, again, that's a relatively sharper transition where the pCNA gets recruited somewhere else and the signal gets lost or almost lost. So this is the trajectory that we're trying to deal with. This is some sort of data that we're trying to annotate. And so the first thing that I did, I was a PhD student. What could I do? I just manually annotated these trajectories. And the problem with the manual annotations was that no matter how hard I tried, I could never tell where the transition between the S phase and the G2 phase occurred. And frankly, if I asked, I don't know, the audience here, and if I asked every single one of you, where do you think that transition occurs? Where would you pinpoint it to the single frame? Some people would say like, oh yeah, it's exactly there. And someone would say it's the next frame because, that's where the speckles start occurring. But frankly, you would have some uncertainty in the model, and you would have some uncertainty in the temporal labeling and what exactly you are trying to do. So we tried, well, okay, instead of, going the classic way of training the classifier, annotating data, putting some labels there and getting the classifier to work and actually measuring its accuracy, we wanted to approach it in a very different way. So what we did here was we kind of like just zoomed out from trying to attach the labels to every single bit of this sequence, and we actually used a different sort of methodology. What I did here was that I took the individual individual crops along my trajectory, and I trained a variational autoencoder to actually compress that image into a smaller latent representations. So for the AI connoisseurs here in the audience, you'll probably know, if you've ever trained an autoencoder, that this is a way of self-supervised learning, because the input image is also your answer. And what you're trying to do is to train a model that can compress the information in your input data and can decompress the information from that compressed representation into the original image. So what you're trying to get to is to minimize the difference between your input and your reconstruction. And the idea behind using the autoencoder is that hopefully by the time that the convolutional encoder compresses the information into the latent representation, that latent representation should be informative enough, and it should be compressing all the key essences of the input data. And this is absolutely great because, as you can see, well, maybe you can't, I don't know, we're dealing with very noisy data here, but hopefully you can believe me that the clusters that are appearing in the input image are actually reconstructed in that reconstruction. The beautiful thing about the autoencoder is that we can kind of visually check for its progress. We can visually check whether the key essence, the clusters and the speckles that we're interested in, whether they're captured in the representation. So that's a fantastic way, because that's a great thing to do, because we can manually validate whether the autoencoder is looking at meaningful results. But as a byproduct of this principle, you get the latent representation. You get this little hypercube here in the center, which is actually a numerical representation of your image. It's a compressed representation. It's not as big as your image was at the beginning in the pixel space. It's usually some compression, which you are free to define. You're free to define how many numbers do you want in that representation. But the fantastic thing is that you can use that representation, and you can believe that representation has all the key characteristics of your image data in it. So, we've done the pCNA image latent representation. We land the representations automatically. Now, how do we proceed from there? So, as you can see, I did deploy this autoencoder along the trajectory, and hopefully you can convince yourselves that all the key essence, or what I mean is all the granules, the presence and absence of PCNA granules in the trajectory is actually present in the reconstruction. And one shout out, I really want to give a shout out to the autoencoder here, because it's dealing with already noisy data, and autoencoders usually struggle with the fact that they add noise onto the data. But you can see that the key essence, the clusters, the speckles, the granules, however you want to call them, they're present, and those phases where they're supposed to be present. So once we do this with the entire sequence, apologies for that. Once we do it with the entire sequence, we all of a sudden get the latent representation of the entire sequence for our data. Now, what can we do with that? It's just some certain numbers. We do not necessarily understand what each number in that representation means. So how can we draw some meaning, and how can we start comparing these sequences one to another? That's the question I'm hopefully going to answer in the next slide. But also, we trained this autoencoder in an unsupervised or self-supervised way. And we wanted to kind of stick to this idea, because annotating sequences and annotating these classes is not going to get us very far, as hopefully you understand why this is hard to do over sequences. So the question that we started to ask at this point was, can we harness the self-supervised approach to annotate cells, to be able to partition them into discrete chunks? And the approach that we used is actually based on dynamic programming. But before I show you what we actually did with the dynamic approach on cells, I'm actually going to show you what we've done. And I'm actually going to demonstrate the principle on two strings. As you hopefully remember, I'm joining you here from the Alan Turing Institute. And so here, for your convenience, I defined two strings for you. As you can tell, as any human, I think, could tell, they encode for exactly the same thing. So you have the Alan Turing Institute strings spelled twice. There's 4 words, exactly 4 words, but there are some local dissimilarities in that sequence which are actually showing you what could be happening in the cell cycle. In the cell cycle, there are certain locations of the sequence of the cells which can be stretched or squished, and they do not have to follow each other in a perfect comparison. So here, what I try to do is to I try to kind of compare these two strings one to another on a pairwise manner. But we actually run into problems, and I tried to motivate these problems before. First of all, the strings are not of equal length. So this is a problem, because by the time that we get to the end of the shorter string, we have nothing to compare these last characters to. The other thing is that if we did the pairwise comparison, you can really see that the pairwise comparison would actually be quite wrong in multitude of places. And this is because the string would actually not be perfectly matched. Even though these two sequences are encoding for exactly the same thing, we wouldn't be able to pairwise get to the perfect matching. So instead of doing this pairwise, maybe we can think of more dynamic or more adaptable approaches to actually get this matching to work. And so this is exactly what I'm showing you here. So again, the same two strings with local stretches and squeezes in the sequence. And what I'm showing you here is that when you use dynamic approaches, you can actually find a perfect matching between the two, just as you can with your eyes. So we actually thought that this dynamic programming and this dynamic approaches could be particularly helpful for the cell cycle, which is full of regions of local stretches and squeezes and alternations in the data. Another way of representing this dynamic matching is representing it in form of a matrix. So here I'm not showing you the entire Alan Turing Institute, just the name Alan Turing, and again it has these local stretches and squeezes in the data. So what I can do is to represent this as a matrix, and I can find the best alignment path between the two strings. As you can see, this is what I'm showing you here in black. So the dynamic approach helps us to actually deal with the two problems that we're facing. First of all, it takes care of the fact that we have unequal global lengths of these sequences, but also it takes care of the fact that there are some local regions of shortening and prolonging of the sequence. This may not seem particularly helpful for the cells, like how is this actually translatable? But let me show you what I actually mean by this. We can use the same string approach, and we can use this dynamic time warping matrix and the best alignment path to actually start inheriting the distances to previously unseen cells. So what I mean by that is that let's now assume that you only know the order of one of those strings. However, you can somehow compute the similarity matrix between the two sequences. What that means is that even for an unknown cell, you can harness the properties of this matrix, and you can harness the properties of the alignment path, and you can start deducing, or you can start descending the labels onto an unannotated sequence. And this is exactly what we're going to do with cells in the next slide. I can compute the dynamic time warping matrix using the latent representations that my autoencoder has learned, and I can start using just one annotated cell. I can start to descend the labels onto a new query which I know nothing about. So let's do it. As I mentioned before, for this approach to work, you need to know at least one labels of the cell. So let's call it a reference cell. The reference cell can be of any length. Let's say that it has 370 frames because we do the imaging quite rapidly. And then we have the query cell that is present for much longer or much shorter. It doesn't have to be equal. That is the point of the comparison here. Then we take the reference cell and the query cell sequences, and we encoding using the VQVAE. With the autoencoder, what is actually happening here is that we get the numerical representation for every single patch in the sequence. And because we know the order, we can actually start mapping and matching the order of these sequences into the dynamic time warping matrix. Now, because we know the labels of the annotated reference, we can harness the properties of the best alignment path, and we can start inferring the phase labels on an unannotated cell. So just to kind of recap all of this process again, sorry, my throat is very dry. So just to kind of recap this process, this is exactly what we can start doing with the cells. And here I'm just showing you an exemplar dynamic time warping matrix without showing you any labels, without showing you any belonging to G1 and S and to G2 phase. You can actually start seeing that there is a lot of regions of local similarity and local dissimilarity in this path. Without understanding what this path mean and how it can be partitioned, you can see that there is internal structuring when we start comparing these sequences one to another. And actually, to understand the shape of these, to understand the shape and the, how do I put it, to understand the trajectory of that path is actually really difficult without knowing anything about the labels. So let's add it in. Now, if I actually understood what is going on in terms of the annotations and which parts are actually belonging to the G1, S, and G2 phase, I can now start inheriting using these labels, and I can inherit them onto an unannotated query. And this is exactly what I'm doing here. And here at the bottom, I'm actually showing you that this is not just a wild guess. When we're trying to do this matching and when we're trying to inherit these sequences, what I can do is that I can contrast it to the best annotation I, as a human, can possibly come up with. And you can see that the IOU agreement, or the agreement of the relative positioning of the G1, S, and G2 phases is relatively cool. You can also see that it's much easier to annotate the G1 phase compared to the S and G2 transition. And the reason for that is because the S and G2 transition might actually be mislabeled by me. I kind of try to motivate the fact that it's really difficult to pinpoint it to the exact frame. So now you may start asking, like, well, okay, so you show this matrix and you show this, IOU agreement between a pair of cells, but how do we trust? How can we believe that this is a reliable thing to do for all of the cells in our repertoire? And that question would be very valid. This is something we started wondering about when we, started gaining some trust into this methodology. And it's, and what I'm going to show you in the next slide is hopefully going to reiterate on that point. So here, what I've done is that instead of just warping a pair of cells, one reference and one query, what I try to do is to actually annotate to the best of my knowledge, a couple of references. Here, I think I'm showing you up to 160 or so, but actually I tried to label a few. And what I tried to do was that I could then take an unknown query, a new cell I knew nothing about, and infer its labels using the dynamic time warping to the 160 cells that I had in my reference library. And what you can see here is exactly the representation that I would get. This is the way I like to think about this is that this is the statistical representations of the cycling phases and the cycling transitions. So you can start seeing that you do not have to trust a pair of cells if you're suspicious that perhaps that pair doesn't capture the transitions really well, but you can start generating the statistical representations and you can start gaining the confidence about which part of your sequence actually belongs to which phase. Now, the beautiful thing about this is that you can actually use this plot and you can transform it into something that I find particularly pleasing, which is the label confidence. And the reason for that, or the motivation for why you would want to do this, is that you would want to be able to express some sort of confidence of how confident your model or our model is to be able to tell the transitions and the relative switching between the phases. So that's exactly what I'm showing you here. The label confidence is frankly just telling the frequency or the percentage of the times that the label has appeared in the above plot. And so you can see that every trajectory, or well, the trajectory I'm showing you here, you got to trust me that it works for on every single one, but the trajectory that I'm showing you here is actually starting at mitosis. It follows through the G1 transition, and it follows through the G1 phase, and all of a sudden it continues into S phase and G2 phase. Hopefully you can recapitulate the fact that the S to G2 transition is actually quite gradual and quite slow, and this is pretty much how I wanted to finish up my talk. But so one thing I actually wanted to say was to actually reiterate on this point that there are transitions which are very difficult to capture. And actually, you can see that this is to the best of my knowledge, this is the transition between the that this is the transition between the phases that I actually annotated as a human. So as you can see, I just tried to kind of open the stage to the fact that perhaps annotating these discrete phases into the in the trajectories may not be the particularly helpful thing, and expressing the confidence can actually help you quantify the uncertainty. Let me just finish with that, and thank you very much to all the collaborators and to your attention.